Ok, buenos días. Nombre es Orla Hardiman de Dublin. Quiero hablar sobre Precision ALS in English. So thank you very much for the invitation to speak about Precision ALS, which is a project that we are running across Europe and focused in Dublin, where I live and work. So I'm going to give a little bit of background. I think this group knows about ALS. We know that it's a degeneration mainly of motor system, with an incidence in Europe of about 2.6 to 3 per 100,000, perhaps a little lower in South America. We know that it's a neurodegenerative condition and overlaps with other neurodegenerations, particularly frontotemporal dementia. And our work and that of others has shown that the rates of familial disease, where we can identify another family member with ALS or frontotemporal dementia, is around 20%. If we include other neuropsychiatric disorders, it's about 30% in the Irish population, perhaps lower in populations such as those in South America, where the population is a little bit more mixed than from where I come from. We know that the disease is complex, and the factors that lead to the development of the disease are complex. We know there's a genetic component, and there is almost certainly an environmental component. And this leads to a great deal of variability or heterogeneity within the disease. And one of the reasons why we set up Precision ALS was really to try and understand this, to explore this variability and heterogeneity to a greater extent so we can develop new treatments. The disease, of course, is uniformly fatal, and we don't really have any effective disease-modifying drugs. When we think about ALS, we think about both the diagnosis and then the clinical assessment. And of course, the diagnosis can be difficult. We know that it takes up to 12 to 18 months to make a correct diagnosis of ALS, and this is fairly uniform across the studies that have evaluated this across the world. Of course, the symptoms can be relatively non-specific, and when patients present to their general practitioner with these types of symptoms, those symptoms are usually not ALS. For example, a foot drop is usually a disc in the back. So general practitioners or non-specialists can be forgiven for not recognizing ALS at its first presentation. We would like to reduce the interval from first presentation to diagnosis. And of course, these are what we would call red flags that we must educate our colleagues to recognize, so that the person can be referred as rapidly as possible to a clinic such as the ones that in which I'm involved, where we have specialists who can recognize the disease to reduce the interval from first symptom to diagnosis. So this is a big challenge everywhere, and we haven't found the answer to this as of yet. We don't have a really good diagnostic test that confirms the diagnosis of ALS as of yet. It still remains a clinical diagnosis. Another aspect of ALS, which we need to recognize, is the presence of cognitive change. And this is not uncommon. Up to 50% of people will have some level of cognitive change, primarily in the executive domains. This is work we did many years ago, back 12 years ago now. We've replicated on a number of occasions looking at the presence of cognitive change, which maps onto the same sort of changes that we see in frontotemporal dementia. We also see behavioral change, and actually it's much more common. Up to 70% of patients with ALS will have apathy as a prominent feature of their disease. And we have a scale which we developed in Ireland called Bowman Behavioural Index, or Bowman Behavioural Inventory, which can map onto different aspects of behavioural change, which in turn map onto different patterns of network disruption within the central nervous system. So ALS is not just a system of upper and lower motor neuron degeneration. It also is a system that's characterized by cognitive and behavioral change. The way we assess ALS is using the clinical outcome measures that have been accepted by the field and accepted by the regulatory authorities. And the prominent one that we use is the ALS-FRS-R, or the ALS Functional Rating Scale. So this is the first 5 points on the ALS scale. It's a 12-point scale in 4 domains. So 24-point scale in 4 different domains. And those domains are reflecting impairment in bulbar function, in gross motor, in fine motor, and in respiratory. We also use respiratory assessments, and these are the main determinants of the outcome measures that we measure in clinical trials. We measure the slope of the ALS Functional Rating Scale, and we measure the decline in vital capacity, or FVC, and we also measure survival. And these are the aspects of the disease that determine, for example, whether a drug is effective or not. We compare the patients on the drug with the control patients. But of course, these scales are neither reliable nor particularly accurate, and there is a recognition now that there are many problems with both the ALS-FRS-R and with respiratory function. We know that the rate of progression is not uniform, even if we look at the slope of the ALS-FRS. This is a figure from Ruben van Eyck's work and published in 2019, showing the variability in the decline of the ALS-FRS. Cognitive change is not measured using the ALS-FRS, so we don't actually have a validated measure of cognitive decline. We use something called the ECAS, the Edinburgh scale, which is developed by Sharon Abrams and her group, and it's quite sensitive to changes in executive impairment in ALS, and there are 3 different subtypes of the ALS of the ECAS, but it's probably not sufficiently sensitive to measure cognitive decline in the context of a clinical trial. So our ways of measuring change in ALS are not very good. We know that there are prognostic factors that determine the rate of change in cognition, particularly executive dysfunction is one of these determinants, although it's not captured by the ALS-FRS. We know that the site of onset, whether it's bulbar or spinal, gender as well, and the rate of decline is measured by the slope of the ALS-FRS. The presence of genomic variants is also a factor, and we've used these to develop the NCAS prediction model, which predicts survival, which of course is partly determined by progression. But the actual progression itself of the ALS-FRS, we don't have a prediction model for that as of yet, but there are a number of studies underway to determine this. Going back to the problems with the ALS Functional Rating Scale, if we take the different subscales within the ALS Functional Rating Scale, we see that first of all, there are differences in whether the site of onset is bulbar or spinal, but there are also problems in that the scale has floors and ceilings, and you can see that it's not linear. So this is work that James Rooney, who worked in my group, published a few years ago, but other groups have shown this as well. So this is problematic. The scale is not linear in its totality. It's sort of S-shaped, but neither are the subscales linear, and the nature of the onset of the disease, whether it starts in the bulbar regions or whether it starts in the spinal regions, and also the sex of the person will determine this. So there are gender or sex differences, for example, in the respiratory subscores with respect to the rate of decline. So even though we use this scale as an outcome measure in clinical trials, it is far from perfect, and this is a problem. There are also some reliability problems with the ALS Functional Rating Scale. So this is a study published by Chris McDermott in the ALS journal, which I edit, showing differences in the scale, variability within the scale, coming from sites that are very experienced in engaging in clinical trials. So these are the number of patients with a 5-point or more increase, which should not occur in the ALS-FRS total score. So the scale is problematic, and we need to find better ways of measuring change over time in ALS. So I'm going to come to that later in my talk about what we're trying to do to resolve this. Another problem that we have is understanding the causes of ALS. And we know, as I mentioned, that ALS is probably a gene-environment interaction. And this is a paper that M.R.L. Shalaby published in Lancet Neurology in 2015, considering that ALS is probably a 6.6, there's 6 factors that occur which lead to the development of the disease. Now, the mathematics and statistical modeling behind this have been disputed quite recently. But I think in terms of thinking about the disease as a genetic predisposition coupled with environmental exposures is still valid. It's still valid to think about this as a gene environment exposure. And of course, we've been looking at the genetic profile of those who develop ALS and to understand what the genetic factors that might lead to the disease in the presence of these genetic factors, whether additional events might occur and what those events might be. And this is the 6-stage hypothesis. So we know, for example, that if we carry a variant in SOD1, the steps reduced to 3. If we carry a variant in C9orf72, the steps reduced to about 4. So you still need some environmental or subsequent exposures in order for the disease to manifest, even in the presence of a significant variant that is disease causing. And understanding what that might be, or what those additional events might be, or whether there are additional genetic factors that interact, remains to be established in a very conclusive manner. We know that one of the risks of developing ALS is heritability. So we know that the presence of genetic signals, not single genes, but probably multiple genes with small effect, account for about 50% of the variability in the risk of developing ALS at population level. So this may differ from individual to individual. This is a paper we published a couple of years ago using the Irish population-based register and showing that, in fact, there's a slightly different heritability risk in the single genes of major effect, particularly if the transmission is from female rather than from male. So the liability threshold, the threshold of developing disease, is a little bit higher in women. So if women carry the disease, they're more likely to transmit that risk to their offspring. So this is something that requires additional work. The sex effect of the genomic effect of sex is not well understood and is a subject of quite a lot of investigation now, because this may modulate the risk. We know that ALS is commoner in men than it is in women. The age of onset is slightly older in women, but in heritability, the female transmission is more likely to transmit the disease. So what the underlying biological genomic processes that are occurring to explain this are not well understood at the present, but there are groups, including our own group, that are looking at this. With respect to the single genes of major effect, there's at least 40 now. This is quite an old slide from 4 years ago. But in reality, at population level, the SOD1 was the first we described, and it accounts for a significant, maybe 1 to 3% of sporadic, and that between 10 and 20% of familial ALS. Within the US, it's less in Europe. In Ireland, we have no SOD1 mutations. Familial in Italy, it counts for about 13% of familial ALS, and so they're different countries. Sorry, my little circles are hitting the wrong gene. C9orf72, of course, is the most common, and that circle should be on FOS, and TDP43 is the fourth. So these are the genes that are most important. My circles are not hitting the right place. So we know these 4 genes are the genes that are most important in populations of European extraction. But of course, this is different in other populations. C9orf72 is really not very common at all in Asian populations, whereas SOD1 is. In South American populations, C9orf72 is less common depending on the population that we look at. Of course, in Argentina, much of the Argentinian population is of Southern European extraction, and the rates of C9 are lower in those populations. Compared to Northern Europe, we know C9 off is there's a founder in Northern Europe up around Finland and the northern part of Europe. We know that there's an overlap between ALS and FTD, and we can demonstrate this genetically as well. There are certain genes that can lead to both ALS and FTD, C9 off, 72 being the most common. There are genes where we don't see very much in the way of cognitive change, for example, in SOD1. And this also relates to the type of deposition that we see. Most of ALS has TDP43 deposition, but there are rare types of ALS, for example, SOD1, that doesn't carry the TDP43 deposition, similarly for FUS, the FUS variant. From the pathobiologic understanding, most of the work has been generated in models, and particularly in the SOD1 model, which was the first animal model of ALS, and that led to development of a very large number of clinical trials that were initially initiated in the animal model and then brought to human disease unfortunately, without any great benefit. And there are many reasons for that, but the reality of the situation is that all of these drugs failed in either in phase 1 or phase 2, or some in phase 3, very expensive, over 100 trials. The trials, the point of early phase 1 and phase 2 to phase 3 is called the Valley of Death, where we might see a signal in an early small number of patients, but then when we bring the study to a larger group of patients, we can show that the drug isn't effective. And we've seen this most recently in the Amylyx compound, where the study, the Senator study in the US, small number of patients, was positive, whereas the Phoenix study in Europe has been negative, showing fairly conclusively that Relyvrio, which is the Amylyx compound, doesn't work. And this also happened with the Darvone small study in Japan, licensing the drug in the US, and then a larger study called the ADORE study in Europe, where the drug was shown to be ineffective. So we've not been doing very well in the provision of new drugs that have been successful in animals, and bringing these to humans has not been effective. And there are many reasons for this. Some of these relate to problems within the preclinical modeling, that the extent to which that's translatable to humans, and see these are some of the factors here. The human problems, one is that the drug maybe didn't work, but there are much more complicated problems that we need to really grapple with. Many treatment paradigms are designed based on the factors confer risk, but of course the progression of disease is a different pathogenic process, and we need to be mindful of this. Of course, there are many trials, earlier trials, that probably weren't very well designed. Many trials where it wasn't clear that the drug did what it was supposed to do, may not have got into the central nervous system. These are the much earlier trials. Now our trials have biomarkers to demonstrate pharmacodynamic effects, that the drug is actually doing what we want it to do. It's also the case that the effect of the drug may be confined to subgroups of patients, and that's something that we really need to consider. ALS is a heterogeneous disease. But really, one of the things that we need to consider is that the modeling of the disease in the fly or the mouse will only bring us so far, and that our understanding of human disease is really lacking, and we need to do a lot more work to understand the disease in humans. Effectively, the best model of this disease is in humans, because it is a human disease, and there isn't a perfect model of ALS in animals. It doesn't exist naturally in animals. So in relation to ALS, we have to think about this as a syndrome rather than a singular condition. This is what's driving the conceptual framework of precision ALS. Even familial disease, not all familial disease is the same. There are different pathogenic mechanisms and different subphenotypes. And as I mentioned, the risk for developing ALS may differ from the factors that drive disease progression. And this really should determine how we target treatments, but this is not always the case. We focus on what appear to be the genomic basis to cause the disease, but these diseases don't manifest till much later in life. So there are things that we need to understand to a greater extent. The disease onset may be just, it's whether we can determine it clinically. So it's an arbitrary clinical definition bounded by the phenomenology of what we can determine as clinicians. But the processes that lead to this within the neuroaxis we are blind to, we cannot see these. And so we need to identify mechanisms by which we can interrogate this process. So really, we don't understand the underlying biological processes that lead to disease onset, nor do we really understand the processes that drive variability in clinical progression, or even in clinical presentation. And these are the things that we are trying to address within Precision ALS. So if we think about the model that Amar generated, and we think about this, we should think about this in terms of risk accruing over time, but the neuroaxis is compensating for this risk. So the risk is developing, but there are factors within the neuroaxis that are able to modify or mitigate the risk. But at some point, the compensation process fails, and then the disease develops, and that becomes irreversible. So where should we target our treatments? We can't really target treatments at risk, except to identify what the risk might be. And this is like, for example, in lung cancer, where we know smoking is a risk, but asking a patient who's developed lung cancer already to stop smoking is pointless because the disease is already manifest. So perhaps we should be thinking about identifying where the compensation is happening but failing, and then maybe intervening at the point of clinical disease or a little bit before that. That's very difficult because by the time the patient presents to us, the disease is already manifest. So the earlier we can detect this, the better. And of course, in the familial forms of disease, we need to be studying this is happening already. Precision medicine is focused on segregating the disease into subtypes that we can recognize, and preferably recognize at a very early stage, and maybe intervene at the earliest point, perhaps at preventing the inexorable decline that we see. So the lessons from where we are from what we have been to where we are today is that we need better and more accurate diagnostic tools. We need to be able to characterize patients better so that we can bring precision medicine-based therapeutics. We need better biomarkers, better outcome measures that demonstrate problems with the LSFRS, better drugs, better ways of developing drugs, and better trial design. And so in order to do this, we've been thinking about this in the European group, the Tricals group. So we developed a roadmap a number of years ago where we described the things that we think we should be doing towards developing new therapeutics for people with ALS. And that's where Precision ALS came from. It's an Irish-based initiative. It's part funded by Science Foundation Ireland. It's an academic industry partnership, and it's with a number of different industry partners and a number of groups both in Ireland, ourselves as clinicians, my colleagues who are data scientists in the ADAPT group in Trinity College Dublin, and the Tricals consortium that is a linked group of centres across Europe who have expertise in ALS. At the moment, there's 10 groups. So Precision ALS, the objectives of Precision ALS is really to develop a system that's going to answer these questions, that really address the lessons that we've learned and the pathway that we need to follow. And really to do this, we need large-scale data collection. And we need to think about this human disease in the same way as we might think about the animal model. So we need to think about this as a very large laboratory experiment with the patient with the disease as the study. And we need to think about this at multi-omic level. We need to think things in terms of clinical deep phenotyping, and then using all of the tools and technologies that we have to try and interrogate the disease so that all of the data that we generate is superior to what I can do as an experienced clinician. And that includes the clinical evaluation that I provide, the imaging studies we can do, advanced neurophysiologic evaluation, looking at the real-time neurophysiologic network changes that are happening. And then all of the markers that we have, the genomic risk markers, but also the markers that we can identify within the body fluids, within blood, urine, and CSF. And that we bring all of these together on a large multi-omic, multi-dimensional data platform that will then have the capability to integrate the data that will allow us to undertake both single modal evaluation, such as we do already, but also multimodal evaluation that are, so the different domains are interoperable, so we can undertake advanced analytics using both traditional statistical tools, but also additional machine-based learning and artificial intelligence-based technologies that will allow us to extract agnostically data and drive the data, have the data drive the analysis, to generate new knowledge, to drive the translation and clinical research in ALS. So that's the purpose or the objectives behind Precision ALS. These are the partners at present. There are 10 sites, actually there's 2 in London now. We are slowly expanding, and we're linked with the European voluntary organization, OIPALS, the umbrella group for the voluntary organizations. And we have a number of different partners, which I'll mention at the end, industry partners that are helping to drive this. So to date, what we've done is we've harmonized many of the data sets that we've generated in the past collaboratively as various consortia. And these are the consortia that I've listed here that have funded research collaborations. So out of those, we've generated a large data set that we're in our own repositories. So what we've done now is tried to bring all of those together in one large data set. We call this the extant data set, with almost 24,000 patients now in that. Now, there's a lot of missing data, but we have sufficient data to enable quite a lot of data mining, some of which is turning out to be extremely interesting. For example, the close to 10,000 patients in whom we have genomic data, about 1,200 of those have a genomic variant. And just looking at the pattern of the variants and how they behave in Europe compared to other parts of the world has been extremely interesting. So we're in the process of publishing this work. It's just about to be submitted. So here's we have 6 or 7 papers looking at the natural history of the NSFRS, looking at survival, looking at time to event, looking at the use of drugs, looking at the impact on day-to-day living, looking at the clinical genetic correlations, looking at how we measure respiratory function and survival, the use of non-invasive ventilation, of which there's a huge amount of variability, and then looking at economic evaluation. These are going to be part of a supplement that we're going to publish within the ALS FTD journal, which will basically be under the banner of ALS in Europe. What do we know from a clinical evaluation point of view? And then we have a number of projects underway looking at different models by which we can predict progression of disease using different statistical models, including Bayesian statistics, using classical Cox proportional models, and using deep learning and artificial intelligence, a number of different ways, a number of different groups looking at this. We're also looking at the cognitive profiling. We have some cognitive data we're collating now, and look at the utility of something like the LS-EQ5 as an outcome measure. So we have some work going on in that. But there are other studies in which we can potentially engage. So the extant data set will continue to grow, and we're open for additional analysis. In terms of what we're collecting prospectively, this has just started now. So our collaborators already have registers or EPRs, electronic patient records. So these are already data that we have, but we're more interested in a more granular data set including the patient journey and the patient experience, which is missing in these registers. So we want to be able to collect the multidimensionality of the disease. And the real world data is very important because it allows us to undertake health technology assessments, in addition to trying to understand the variability and the heterogeneity within the disease. We want to be able to undertake health economic analyses, quality of life and modeling. This is very important for people with the disease as well. And the reason for we accept this, but it's been difficult to collect these types of data, why real world data is important and what the cost is to people with the disease and the carers. So the population-based cohorts that we have that we're collecting are more representative of the entire disease trajectory. We're trying to get the heterogeneity of the disease. So there are existing data sets, for example, the PROACT data set, which is a compendium of previous ALS trials. So it's very useful to model, but only a slice of the disease is captured. Answer ALS is a smallish group of about 1,000. There are a number of other data sets, and there's a new study starting in the US called All ALS. But again, we believe that across Europe, that we'll be able to generate a much more rigorous and inclusive set of patient data that will allow us to really try and tackle this question about disease heterogeneity. So we're using the core elements, and the utility of these data have already been demonstrated. Nobody's lost to follow-up, so we have a lot of data, and ultimately we'll want to be able to generate what you would call digital twins. If we have a large enough data set, we'd be able to model any disease trajectory that we might expect, which would obviate the need for placebo-controlled studies in the future. We're planning to have all the participating sites upload the population-based data. 75% of all incident cases and 25% of all prevalent cases are our targets. We have a milestone-based process, and then the patient journey will collect these data here. The patients are consented locally. The codes are contained locally. In Europe, we have a very stringent general data protection regulation, so we've had to spend a lot of time working to the legal frameworking of this, and this has taken probably 2 years to really have it ready to go. All disease-related activity is recorded. We're going to start collecting biological samples in the near future, but we'll be collecting data from patients at 4-month intervals. So the worksheet has once-off data, which is the patient's demographic and genomic status, and then recurrent sections where we collect information along with. It's just a screenshot of what we're collecting here. So these are our tablets that we've distributed to the sites, and the data are collected electronically and then uploaded on our platform, which is housed in Trinity College. We're monitoring the data collection entry. We'll be auditing the sites on a 6-monthly basis. We'll be cleaning the data. Then we're doing yearly data cuts for analysis. We'll be collating these data with additional biomarker and outcome measures data so that they'll be available for advanced analytics and machine learning. And then we'll be correlating the clinical data that we have with other repositories such as Project Mine and some of the biobank repositories that we're going to have. So as I said, 75% of incident patients, 25% of prevalent patients. So the aim is probably 1,500 is a bit optimistic, maybe 1,400 new patients per annum. So about 4,000 patient journeys after 3 years. So in the longer term, we're expecting about 27,000 European patients by the end of 2026. We're collecting biomarkers. We can think about biomarkers in terms of fluid-based, imaging-based, or near physiologic. And the plan would be to collect in all of these different domains, in all of these categories: diagnostic, categorical, prognostic, and of course pharmacodynamic would be in the context of treatments. But this is the sort of model that we'll be using. So we're collecting imaging. We have imaging data sets. We have a project running with Novartis looking at proteomics. We have some neuroelectric signal analysis underway in a number of different sites. We have some transcriptomics data. And then we have some new work that we're going to be developing around some of the unanswered questions in neurofilaments, and also most recently thinking about innate immunity, PBMCs, peripheral blood mononuclear cells. So these are types of work that we're doing with the neurophysiology, with imaging, with transcriptomics, and with genomics. And we're also developing or working to develop better digital outcome measures to investigate the usability of these novel technologies. So there's a lot of novel technological development out there, but there's huge variability. So one of the objectives we have is to quantify the usability and validity of these technologies, to identify the most sensitive parameters, so that we can use these in tandem with the LSFRS in the first instance to demonstrate their validity, ultimately to replace the LSFRS. So to quantify rates of decline and impairment using these technologies, and then to validate these to provide a unified scale that we can use, that's quantitative, that will supersede the LS/S in time. That's a big ask, but there's a lot of barriers to the use of technological of technologies in ALS, and these are some of the things that we're exploring: what to measure, how to implement, the suitability from a patient and caregiver perspective, and then the stakeholder engagement, particularly regulatory, the users being the stakeholders, also being industry, and also the whether the regulatory authorities would accept these in a clinical trial setting. And then finally, we have a large data science, data analytics part to Precision ALS. So we have a disease machine learning group that is already up and running, that's using some of the extant data to test some of the types of statistical modeling and machine learning technologies that we have available. And going forward, as we bring in more data to look at this cross-modally and multimodally as well. So Precision ALS is a unique academic industry collaboration. This is the website here. I'm happy to take any questions about it. The partners are as listed here at the moment. We're about to expand out into a couple of other sites in Italy, and we're open for business with other sites as well. These partners here are funded in collaboration with Biogen, but as the project grows, the expectation is that we will also be able to onboard additional sites that will be able to upload data. These are the companies that are participating at the moment, but we have engagements with other industry partners as well. You can see that they're mostly pharma companies. Actually, IQVIA is a very important partner here, which I forgot to put up. But we're open for business with other partners as well. So I'll stop sharing now, and thank you for your attention.
Excellent. Thank you so much for that, Orla. That was really interesting and very, very exciting. So yes, it looks like Precision ALS is a program where we say, okay, we have so many wonderful projects around the world. Why don't we put all that data together so we can start nurturing research at global level, right? And there is so much information going around that sometimes it can be overwhelming. So this is the kind of project that in a way comprises all those large data sets that we have around the world. And speaking of that, Orla, you've been involved in LANALS, which is a Latin American network of ALS researchers. So you really have been a big champion in supporting ALS research in our region. And can you share any updates you have on the work of that group?
Yes. First of all, with respect to the first comment that you made, I think that in an ideal world, being able to share data without any difficulty would be very good. The trouble is that I live in Europe, and health-related data is considered sensitive data, and we're subject to the European GDPR, General Data Protection Regulation. So being able to share data across all parts of the research landscape in different countries is challenging for us. It doesn't mean that it's not useful, because, for example, the initiatives that are underway in other parts of the world, we can use each data set as a replication data set for the other data set. As long as we harmonize how we collect data, I think it's okay that we have two different ways, two different jurisdictions of collection, and that we find ways of being able to share the data, but also harmonize what we do and share how we interrogate. So I think that's really important. Regarding the Latin American consortium, I am very embarrassed because I don't speak Spanish, and I think that it's a little bit disrespectful for a non-Spanish speaker to be a leading participant in a study in a language where the native language is different from what the person speaks. So my colleague Monica Povedano from Barcelona has submitted a follow-up proposal to the CDC in the US to continue funding from the US to support the continued development of the LANELS work. So we are still working with the original groups. So that was in Cuba, Uruguay, and Chile. Those are the three original groups. But there's a very large group in Colombia, as you probably know, in Bogota. Of course, there's your own group in Argentina, the number of groups in Brazil. So the idea about the next version of LANELS would be that it would bring in the other centers around Latin America that have a strong research background and that are interested in developing the Latin American consortium. I think within Chile, there are groups that have slightly different perspectives on how this should run. There is some resistance, I believe, and I don't blame them, to have Europeans as helping to lead this. I can understand that. I'm come from the only country in Europe that was colonized, so we understand the perspective of countries who were previously colonized by a European country, but wish to run their own projects now. So we're here to help, and we're hoping that the proposal that Monica submitted to the CDC will be successful, and that that will allow us to continue on with the development of the program. So that's where it sits at the moment. I'm in the background, willing to help, but really it's a bit disrespectful, if the truth be known, for somebody who doesn't speak Spanish to be leading out in this. And for me, it was sensible to ask Monica to become the lead European participant, if that's acceptable to the LANELS group, because she is Spanish speaking. So that's where we're at.
All right. But thank you anyway for all the support that you're giving to the region, and no matter
we're still there. I'm still there.
I'm
still very interested. Yeah.
Whatever your native language is, it's very important for developing countries to have the support from those countries where the main line of ALS research happens, right? So you speaking about different data sets around the world, you mentioned in one of your slides Project Mine, and we remember like 10 years ago or 7 or 8 years ago, everyone was talking about Project Mine, which is a huge project at global scale. but we haven't heard that much about this project in the last few years.
Yeah.
What can you tell us about this specific project?
So Project Mine is still publishing. The data set is quite large, as you say. I think it's about 22,000 samples now. So it's about, I think, 15 or 16,000 ALS patients and about 7,000 controls. The cost of whole genome sequencing is much reduced now. When we started in Project Mine, it was over $1,000. Now it's about $250 to sequence. So it's still running. From a genetics point of view, the geneticists feel they have scale now to undertake some of the analysis that they wished to undertake. But I think if people want to contribute, which we are still contributing in Ireland, I think the Project Mine is open for business, and it's been run by Jan Veldink in the Netherlands. My group, Russell McLaughlin, Russell McLaughlin is PI in his own right now, but he works with me still. So he's leading out from the Irish part of this. And there are a number of different subgroups within Project Mine looking at different aspects of genomics. So there's a epigenomics, there's a transcriptomics, there's a phenotype subgroup. So they're undertaking quite a number of different analyses and publishing extensively. There's still quite a lot of work coming out from the Project Mine group and a lot of interrogation of the data set. We have some projects that we want to develop within Project Mine as well. So, for example, the C9orf72 variant, which is a very important genomic factor in some forms of ALS. About 10% of ALS in Ireland carry the C9orf72 variant, but actually we've identified a number of families in Ireland where some members of the family with ALS have the variant and some members who have ALS don't have the variant. So one of the things that we want to do in Project Mine is take out all of the C9 families, all the C9 positives, and look to see how many of those have family members who also have ALS. So there's quite a lot of that sort of work we can do in phenotype, genotype, characterization. So that's one simple one, but there are many other parts of Project Mine that are underway. The epigenomics one in particular is very important, looking at the factors that might, the other factors, environmental perspective, might have led to the incurring risk of disease. There's a lot of association work underway looking at genes that may be important in the cause of the disease, and then another set of genes that are important in the progression of the disease. So some publications on that as well. So it's still there. It's probably not as very much in the limelight because the collection processes probably aren't as driven as they were, but the data are being analyzed, and we'll be linking our data with the Project Mine data as well. So some of the Project Mine phenotypic data probably could be a lot richer than it is, and we'll be helping with that in Precision ALS.
Excellent. Thank you for that. So as you well know, and actually you also mentioned this during your presentation, we've been having our ups and downs in our ALS research landscape lately, right? So what's your stance on all these conditional approvals and the tendency of making trials as rapid and as short as possible?
So I think there's been a big disconnect between the northern part of America and Europe in this regard, and the drivers for this are different, and we understand that. I'm not so sure what it's like in the southern part of the Americas, but in the northern part, the patient voice is very loud, and the this has driven my colleagues in the US and also the regulatory authorities in a way that perhaps is not not particularly good in the longer term. So my view is that a phase 2 trial should be followed by a phase 3 trial, and 6 months is too short for a phase 3 trial. In Europe, the Amynex sought conditional approval in Europe by EMA for their drug and were refused. And then about 3 months later, the Phoenix study proved that the drug was ineffective within the cohort that we were enrolled. So my view is that we need to be cautious. We need longer trials than 6 months. We need larger groups of patients. The LSFRS is an outcome measure that we need to use for the next couple of years, but we need to recognize the limitations of it. And I think conditional approvals are probably not a very good idea, and they're not really available in Europe anyway. Does that answer your question?
Thank you. Yes, you did answer the question. Thank you for that. But on the bright side, we had tofersen and this antisense technology that seems to bring a lot of hopes for the future of ALS research, and even the possibility of preventing ALS. So what is your opinion about that?
I think the proof of concept that's been generated by tofersen is wonderful, but I think in a way it was the easiest to use. It was the easiest model because when we look at the other ASO that has reported on C9orf72, we've had 2 studies now that are negative. So I think there's a bit of a road to travel yet, and this goes back to my idea or my conjecture that there's a compensating risk part to the disease. Then there's the risk is no longer mitigated, and then the disease becomes inexorable. So for SOD1, we know it's a toxic gain of function. And we know that if we turn off the SOD1 gene, it looks like we can slow the progression. And this is probably also the case with FUS, with FUS. But I think that's a long way to go before we can think about this in other types of ALS. So I think it's certainly gives us hope. But I think that there are also aspects of the disease that we don't look at properly, or that we don't really understand. And one aspect is the human neuroaxis, and understanding what's happening not just at single cell level, but at the level of the brain networking. And it may be that drugs that work at that level may be able to help to maintain the compensatory process. So not necessarily ASOs, but maybe either drugs that work across multiple different receptors, or modulatory receptors, or multiple drugs that are working at different parts of the neuroaxis. So I think there's a risk if we focus exclusively on ASOs, we may be guilty of tunnel vision. And that we need to think about this as a really complicated disease of humans, where there's a spread process, but the spread process includes the disintegration, or the disruption and disintegration of brain networking. And if we can try and tackle the disease at that level as well, that may not be at the level of an ASO, it may be at the level of multiple different modulatory processes to maintain the compensatory mechanism that's underway the first half of people's lives before they develop the disease. So I think there's a lot that we don't understand yet. And I think looking at the disease in both at a single cell level, at an environment level within the neuroaxis, so looking at the innate immunity processes, and also then looking at the way the disease is spreading at the level of the network. I think all of those parts of the disease we need to study. And the latter two are very difficult to study in vitro. The modeling that we use in the IPS model at the organoid level is not sufficient to study those two aspects. So I think there's quite a lot of additional work that we need to think about and be clever about the way we look at the human disease in the sorts of modalities that I talked about. So a lot of interesting work going on at innate immunity level, looking at things like T regulatory cells. This is coming originally from Stan Appel's work from many years ago, but there's some very nice work that Andrea Malaspina in UCL and Caroline Ingrid in Stockholm have been looking at T cell subtypes, and Andrea has been thinking about T cell senescence and the loss of regulatory innate immune regulation, which may be one of the factors that leads to the triggering of the disease. So I think ASOs are one part of the armamentarium, one part of the strategy towards treatment, but I think we need to have a much better understanding of the entirety of the disease in the human before we think about this as the only approach. We'll be seeing the readout for Ataxin 2 in the near future. It's going to be presented at NCALS, and that will be interesting. That's a phase 2 study. So if that's negative, that's another ASO that didn't work. We don't know yet what it shows. So maybe if it's positive, that'll be very good. We have the Statman 2 study that's supported by Curalis. That's an ASO as well. So that's on hold at the moment. We're hoping that the next cohort will be enrolled in the near future. So it's one target, it's one strategy. My worry is that if the pharma industry decides that that's the place where the money is going to go, my worry is that we'll lose out on the wider type analysis that we need to do in the disease, in the human, for us to understand and develop a more wide-ranging set of potential treatments for the disease. And I think an analogy would be, for example, cancer therapeutics. Even though there's been a development in our understanding of cancer therapeutics, the understanding this and using multiple cocktails of drugs in addition to modulating the immune system has been a successful approach in cancer. And I think looking at this from a neurodegenerative point of view is probably the way that we should be thinking about as well. It's much more complicated than targeting one pathway, in my opinion.
Okay, Orla, thank you so much for all that. It was a very interesting talk. It was an excellent presentation, and thank you for addressing all of our questions. Thank you for keep contributing for our ALS environment and in all the world, not only in Ireland and in Europe. And it's always good to see you. We've known you for a while. We've been seeing each other in different meetings, so we are looking forward to seeing you again next time, probably in Montreal.
Yes.
I don't know. And so thanks again for taking.
Thank you very much for the invitation. I really
taking some time of your very busy schedule to do this presentation. Everyone in the ALS Association of Argentina says hi, so they are sending their greetings, Dario and all the people in our association. Okay.
Thank you very much. Thank you for the invitation.
So un gran abrazo para ti, Orla, y muchas gracias por esta presentación. Thank you so much.
Gracias. Thank you.
Take care. Bye.